Hello everyone. So in this topic, I'm going to highlight on the microbial photosynthesis. So in all of the metabolic pathways that you've learned, that we've just discussed earlier on, we know that organisms obtain energy for cellular work by oxidizing organic compounds, right? So about where do organisms obtain these organic compounds? So some, including animals and many microbes, feed on matter produced by other organisms. For example, bacteria may catabolize compounds from dead plants or animals, or maybe obtain nourishment from a living host, right? The parasitism. Other organisms synthesize complex organic compounds from simple inorganic substances. The major mechanism for such synthesis is a process called photosynthesis, which is used by plants and many microbes. All organisms that can do photosynthesis is called a photosynthetic autotroph. So these autotrophs are able to generate their own organic matter through photosynthesis. Alright, so what is photosynthesis process? It is the conversion of light energy from the sun into the chemical energy. The process of photosynthesis can be summarized with the following equation. Plants, algae, and cyanobacteria use water as a hydrogen donor, which then convert carbon dioxide and light energy to the organic matter. Organic matters mean the glucose, as well as convert the carbon dioxide into oxygen. So whereas purple sulfur and green sulfur bacteria use H2S as a hydrogen donor, producing hydrogen granules. So for this type of bacteria, they convert light energy and carbon dioxide into glucose, as well as the sulfur granules. So in this reaction, there's no oxygen is produced. There are two types of bacteria that can do photosynthesis. Number one, the photoautotroph, such as plant, algae, and cyanobacteria. Number two, the photoheterotroph, such as purple sulfur, green sulfur, purple non-sulfur, and green non-sulfur bacteria. So what are the differences between these two groups? So the photoautotroph, the keyword autotroph here, so as you have learned before, autotroph use carbon dioxide, all right? And then in the process, they release oxygen. But for the heterotroph, instead of carbon dioxide, they use organic compound, such as H2S. But in the process, they do not release oxygen. So you can say that photoautotroph is the oxygenic photosynthesis, and then photoheterotroph will go through an oxygenic photosynthesis, depending on whether they produce oxygen or not produce oxygen. Now, you know there are two types of photosynthetic bacteria. They are either the oxygenic or the anoxygenic bacteria. The oxygenic bacteria are those bacteria that uses water, H2O, as the electron donor in the process of photosynthesis. For example, there are cyanobacteria or algae, and of course plant. Okay, but for the anoxygenic bacteria, so some bacteria can use light energy to extract electrons from molecules other than water. So the molecules other than water, for example, H2S, hydrogen sulfur, and then also organic electron as organic compound as the electron donor. So these anoxygenic organism or bacteria occur in the domain bacteria that have representatives in 4 phyla. So which are the purple sulfur bacteria, purple non-sulfur, green sulfur bacteria, and the green non-sulfur bacteria. The anoxygenic photosynthesis depends on the electron donors, as I mentioned before, such as the sulfur compounds molecules, molecular hydrogen, or the organic compound. So these bacteria, these anoxygenic bacteria are normally found in fresh water, brackish water, marine, and the hypersaline water. The bacteria, the group have been divided. So this group is divided actually based on the pigmentation. So you have the purple bacteria or the green bacteria, as well as the third group is the Heliobacter. Purple bacteria are not always purple in color. All right, so they are also brown, pink, brown, red, or violet color based on the carotenoid content. Um, the first group is the purple sulfur bacteria. Okay, they are in the family of Chromaticeae. They are gram-negative. They have bacterial chlorophyll A and B, chemoorganotrophs that utilize acetate, pyruvate, and others. They normally have bigger in size compared to the green bacteria because in the cells they are packed with the sulfide, intracellular sulfide deposition. Alright, so normally these purple sulfur bacteria are found in the anoxic zone of lakes and sulfur springs, so no oxygen. Alright, so examples of these type of bacteria are the Ectotheorodospira, Chromatium, Theoscapsa, Theospirillum, Theodictyon, and Theopedia. So the second type is the purple non-sulfur bacteria. So originally, scientists thought that these bacteria do not use sulfide, okay, but in fact, they actually use a very low concentration of sulfide. But the concentration of sulfide utilized by the purple sulfur bacteria proved toxic to the non-sulfur bacteria. So, so earlier scientists thought that these bacteria are unable to use sulfide as electron donor for the reduction of carbon dioxide. So therefore, they are named as the non-sulfur bacteria. So they are most versatile energetically because they are either photoorganotroph or chemoorganotroph. So they can use a lot of different sources of carbon such as organic acid, amino acids, benzoate, and ethanol. But also these bacteria needs vitamins to grow. The examples of the bacteria are Rhodomicrobium, Rhodopseudomonas, Rhodospirillum, and Rhodocyclus. So the third phyla is actually the green bacteria. So in the color again, so it is not necessarily green. So these are actually the color of these bacteria is actually brown. So due to the presence of the carotenoid compound, so they are gram-negative and contain a different type of bacterial chlorophyll. So they contain C, bacterial chlorophyll C, D, and E. Also a small amount of bacterial chlorophyll A. The important photosynthetic apparatus in this green bacteria is chlorosome. So you're going to look at the structure later. So this green bacteria does not require vitamins for their growth, and also green bacteria has two types, the green sulfur and the green non-sulfur bacteria. The green sulfur bacteria are in the family of Chlorobiaceae. They are non-motile, okay, rods, spiral, and coccile. So they can have different shape of their structure of their cells, and then the chlorosome present in the cells. They are strictly anaerobic and obligate phototroph. They need light. All right, they deposit sulfur extracellularly instead of in the intracellular like the purple sulfur bacteria. Therefore, the size is smaller for this green bacteria. All right, examples of these bacteria are Chlorobium, Prosteochloris, Pelodictyon, and Chlorofertin. Green non-sulfur bacteria is a motile bacteria. So filamentous, the gliding bacteria, they are thermophilic in nature. They can grow at high temperature. So the pigments are bacterial chlorophyll A, C, and carotenes. Chlorosomes are present when grown anaerobically. They are photoheterotrophic and photoautotrophic and show gliding movement. They do not deposit sulfur, therefore non-sulfur bacteria, and examples is chloroflexes. The final group of the N-oxygenic photosynthetic bacteria is the Heliobacter. Based on the 16S RNA sequencing and other morphological and biochemical characteristics, Heliobacter are very different, are quite different with other N-oxygenic photosynthetic bacteria. So they are gram-positive, rod-shaped, and motile. Examples of these bacteria are Heliobacterium, Helophyllum, Helobacillus. Very easy to know that these bacteria are the type of Heliobacter. So they mostly produce endospores, grow up to 42 degrees. They have green in color. So they are green, okay? And so you can find them in the tropical soil or paddy fields. And they have bacterial chlorophyll G, okay? Pigment bacterial chlorophyll G. So I have introduced to you all about the N-oxygenic photosynthetic bacteria. So now it's about the oxygenic photosynthetic bacteria. So these bacteria are unicellular or multicellular. They possess bacterial chlorophyll A and they carry out the oxygenic photosynthesis. They are mostly represented by gram-negative cyanobacteria, okay, and they have carboxysomes and gas vesicles, and they can move by gliding movement. All right, so the process of photosynthesis are oxygenic, producing oxygen, and they are autotrophic, so uses carbon dioxide only. So in here are actually these summary of the different pigments that the different groups of bacteria has that I've mentioned earlier in the earlier slides, as well as the type of the electron donor. All right. So for the anoxygenic bacteria, so you can see that all the pigments are called bacterial chlorophyll, whereby for the oxygenic bacteria, cyanobacteria use chlorophyll, okay, chlorophyll D or F. One of the important components in photosynthesis that bacteria should have is actually the photosynthetic pigment. Carotenoids is important in all photosynthetic tissues because of their role in absorbing the light energy and then transferring it to the chlorophyll or bacterial chlorophyll to be utilized in the photosynthesis process. Cells that are rich in carotenoids but do not have chlorophyll cannot actually do or perform photosynthesis. So chlorophyll and carotenoid are important. The photosynthetic organisms contain either chlorophyll for the oxygenic phototroph and bacterial chlorophyll for the anoxygenic phototroph. Chlorophyll is green in color because it absorbs the red and uh, blue light preferentially and transmit the green light. So what we see is green in color. The absorption spectrum of cells containing chlorophyll A shows a strong absorption of red light with maximum absorption at the wavelength of 680 nanometer, and the blue light, which is the maximum at the 430 nanometer. So this shows the different types of pigment, chlorophyll A and B, and their absorption spectra. So where is it at the maximum absorption? So other than bacterial chlorophyll and chlorophyll are important pigments for photosynthesis. So other pigments I've mentioned before, carotenoids. So these are important to capture and process the light energy. So carotenoid will absorb light in the blue region, but the organism will appear yellow, red, brown, or green. Okay. The second pigment is phycobilins. So phycobilin can be either phycoerythrin that have red pigment, so and then phycocyanin, they are in the blue pigment. Question. So why do different phototrophs have different forms of chlorophyll or bacterial chlorophyll that absorbs light of different wavelength? So this is actually to allow the different phototrophs to make better use of the available energy in the electromagnetic spectrum. Uh, Only light energy that is absorbed is useful for energy conservation. So by having different pigments with different absorption properties, different phototrophs can coexist in the same habitat. So each organism using the wavelength of light that others are not using. So thus, pigment diversity has ecological significance for the useful coexistence of different phototrophs in the same habitat. Prokaryotes do not contain chloroplasts, so their photosynthetic pigments are integrated into the internal membrane system. So these systems arise either depending on the species or depending on the group or the bacteria. So number one, it can be from the invagination of the cytoplasmic membrane. For example, in the purple bacteria, from the bacterial cytoplasmic membrane itself, for heliobacteria, um, and then in both the cytoplasmic membrane and a specialized structure enclosed in a non-unit membrane called chlorosome, so specifically for green bacteria, or um, or the pigments are actually in the thylakoid membrane, for example, in the cyanobacteria. So this is how it looks like for the eukaryotes. So where does this photosynthetic assembly in the chloroplast? All right, so they are attached to the thylakoid membranes. All right, for this is algae. Okay, for prokaryotes, so this is a figure showing the photosynthetic pigments, the bacterial chlorophyll, which is integrated into the a structure called chlorosome. So this is in green bacteria. So the chlorosome is attached to the actually to the membrane, cytoplasmic membrane. Okay, so this is where all the pigments are attached to, and then this pigment will actually absorb light and then proceed with the photosynthetic reaction. Photosynthesis occurs in two stages. So the first stage is the light-dependent reactions, so or called as light reaction or the Hill reactions. So in this stage, light energy is captured and it is being used to make energy storage molecules, which are ATP and NADPH. In the second stage is the light independent reaction, so we don't need light. Okay, so during this stage, so the end product from the first stage, ATP and NADPH, is then used to reduce carbon dioxide. So this is called the Calvin cycle. So Robert Hill is an important person uh, in relation to the photosynthesis findings. So he found that, or he demonstrated that during the photosynthesis, water is the source of electrons in the light reactions and not the carbon dioxide. So in the photosynthesis process, water is important for the electron in the light reaction, but not the carbon dioxide. But this is correct for plant, but for bacteria, um, so they use other than water. So bacteria need hydrogen or hydrogen sulfide or ammonia as the source of electron in the light reactions. So let's have a look at the process of the photosynthesis, um, which involve two steps. The first step is the light reaction. So we're talking about the light reactions here. So in the light reactions, so ATP and NADPH are produced. Okay, so this is what happened in the algae Chlamydomonas. All right, so the event happens are firstly the light that being absorbed will activate the photosystem 2, all right, and then it sets up a chain of reactions in which electrons are released from the chlorophyll. And then this electron is then transported along the chain of carriers to photosystem 1. Okay, so the empty position in photosystem 1, photosystem 2 is then replenished by photolysis. So photolysis will produce water. Sorry, photolysis of water. So this photolysis will produce oxygen and hydrogen ions. So these hydrogen ions, okay, the concentrated hydrogen ion will be pumped out into the interior of the granule will produce ATP. Okay, the final electron and the hydrogen ion acceptor is NADP, and which receive this from the photosystem 1. And then finally in the first step, so NADPH and ATP is produced, and then these two molecules are then fed into the stroma for the Calvin cycle. So now let's move on to the second stage of the photosynthesis process. So in the second stage, it is called as the light independent reaction, so or also known as the Calvin cycle. In this Calvin cycle, the product from the first stage, which are the ATP and NADPH, are then used to reduce the carbon dioxide. There are three phases in the Calvin cycle, which are the first is the carboxylation phase, and then you have the reduction phase, and then we have the regeneration phase. So this is the pathway for the Calvin cycle. Okay, not Krebs cycle, but Calvin cycle for photosynthesis. The second stage, the second phase of the photosynthesis. So you can see here there are three phases, which are the carbon fixation phase, and then we have the second phase, the reduction, and then the third phase is the regeneration of the ribulose. Okay, so in the first phase, the carbon fixation phase, a molecule of carbon dioxide here from the air, from the environment, is being fixed by the bacteria. So this carbon dioxide will combine with a 5-carbon acceptor molecule, which is here is the ribulose 1,5-biphosphate, or RUBP. So when it is fixed, then this step will make a 6-carbon compound. 6-carbon compound, which is actually then splits into 2 molecules of 3-phosphoglycerate. So this reaction is catalyzed by the enzyme called Rubisco, or ribulose biphosphate carboxylase or oxygenase. Okay, so that is the first phase. In the second phase, the reduction, ATP and NADPH are used to convert the 3-phosphoglycerate into molecules of 3-carbon sugar, which is the glyceraldehyde triphosphate. Okay, so this is a stepwise reaction. The first one, 3-phosphoglycerate is converted into 1,3-biphosphoglycerate, and then converted into the G3P, the glyceraldehyde triphosphate. So this stage gets its name reduction because NADPH donates electron to or reduces a 3-carbon intermediate to make glyceraldehyde triphosphate. All right, the final phase, which is the regeneration of ribulose. So some of the glyceraldehyde triphosphate molecules will actually go to make glucose, which is a product for the photosynthesis, while others will be recycled to regenerate the ribulose biphosphate, the acceptor of the carbon dioxide. So in order to make the ribulose 1,5-biphosphate, so it requires a very complex network here. Okay, so here it's not explained. So it is important to know that what is the aim for this Kelvin cycle, what does it achieve, and what does it what are the intermediate that is involved, and then what is the product. Okay, so the main point that you need to remember in this phase is that the carbon dioxide is being fixed to and then being converted into the glucose, right? The product of the photosynthesis. Important point that you need to remember for this Kelvin cycle is the carboxylation phase, which involves the enzyme called rubisco or ribulose 1,5-biphosphate carboxylase, and this rubisco enzyme catalyzes the addition of the carbon dioxide to the 5-carbon molecule, which is the RUBP, and then form 3-phosphoglycerate. So, and then of course producing glucose. All right, so that is actually the complete process of the photosynthesis, the light reaction, and then in the end the independent light reaction. The light reaction will produce ATP and NADPH, and then in the second phase, the light independent reaction, the Kelvin cycle, will use that ATP and NADPH to reduce the carbon dioxide to form glucose at the end. Alright, so that's all for the photosynthesis topic. So you need to remember what are the types of the bacteria that can perform photosynthesis. You know, you have the oxygenic and the anoxygenic bacteria. Also, the different pigments of photosynthesis that the bacteria has. The bacteria have in order to absorb light and then convert the light energy into chemical energy. Or also the final step is the photosynthesis process, which I just explained just now, that involve the two phases. Alright, so that's all for the photosynthesis topic. Good luck for your exam.